Today we will be covering cellular metabolism and some thermodynamics. So we will begin with the thermodynamics portion. For some it may be a review, and for others it may be a new concept. Right? So what we have here is a activation energy chart. Okay, so we have some sort of reaction going on, and we run it to completion, and we find out that our reactants, let's give it an imaginary number. Let's say that it was about 5 calories right here of energy, and then whenever the reaction completed, it was down to 1 calorie of energy, let's just say. So we know that the reactants had a higher energy and the products have a lower energy. Now this is actually favorable. Okay, why is it favorable? Well, to understand why it's favorable, we have to go into the concept of thermodynamics, specifically Gibbs free energy. Okay, so this is going to be Gibbs free energy. Okay, and we will denote it as the rate of change, which is delta or baby triangle, and we will have a G right there for Gibbs. Okay, so this is free energy. And what is important is that if this is negative, so if negative, then the energy decreased, and that is favorable. So energy decreased, and this is favorable. Because really, your body is kind of lazy. It doesn't want to absorb more energy, because when it absorbs more energy, it takes, it's weird, but it takes more energy to keep it up, right? So you always want to be in a relaxed state. Whenever the universe is doing reactions, it doesn't want to absorb energy. It actually wants to release it. It wants to go to a less state of energy. Okay, so this is favorable if it's a negative number. So for instance, like -1, or if the value decreased. Okay, so if you have 10 calories of energy, if it goes down to 5 calories, that's good. You actually released energy. Okay, but what if delta G is positive? So what if that happens? So if positive, you actually absorbed energy, and that is not favorable. Okay, the energy, so we could say the energy increased. So if we look over here, we're gonna kind of do like a copy. Here's our reactants. If I have my transition state right here and it ends, notice that relative to the starting point, the end point is a lot higher. That means that the energy actually increased. So if we have 5 calories over here, we're gonna have about 10 calories right there. So this is really bad, okay? You do not want to be absorbing energy during reactions. So we have these cool words that we call these. If you lose energy, this is gonna be what? Exergonic. This is exergonic, okay? Exer sounds like exit, so energy is exiting the system. Okay, so that's exergonic. And over here, we can also have endergonic. So ender sounds like enter, right? And well, it sounds like it. So endergonic means that you actually absorbed energy. Okay, you absorbed energy, and it's pretty. If you want to have an example of this, whenever you have a fire extinguisher, that's an endergonic reaction. Okay, you pull the trigger and you release the carbon dioxide. It's very cold. It's going to be absorbing energy. So the carbon dioxide, which is a foam in this case, or like a mist, absorbs the energy from the fire, suffocates it, and that is an endergonic reaction. Okay, so whenever we have this happening, we can say that this is spontaneous. And this is non-spontaneous. Right? So spontaneous mean it doesn't mean, oh, it just happens instantly, within 5 milliseconds. No, spontaneous means that, oh, it's going to happen eventually. You don't have to put your own energy into the system for it to get going, right? So for instance, if you have a car, you're going to have to put energy in the form of gasoline. The car is not going to move by itself, right? It is a non-spontaneous entity, right? But if you have a car that is solar powered, well, you don't have to put energy in the form of gasoline, right? So it is a spontaneous reaction. It just uses energy from the sun. So those are my examples for that. Okay, so we can now introduce the concept of the standard free energy. So that is, again, baby triangle. It's also a delta. We have Gibbs right here, and then we can have the little circle. Okay, so what is the standard free energy? Well, it's essentially it's Gibbs free energy, but measured under certain conditions. Okay, so the scientists were like, all right, well, if we're going to do experiments, we're going to have to measure Gibbs free energy within some standard conditions, right? So we can't be changing these things. Kind of like standard room temperature is like 25 degrees Celsius. It has the same thing. So standard Gibbs free energy is about 1 atmosphere of pressure. So it's just the atmosphere we're used to. So 1 atmosphere of pressure. It is at 25 degrees Celsius. 1 molar of solution. So if you have a solution of proteins or something, you want to measure the standard free energy, you have to make sure your proteins are at 1 molar. Okay? And the pH is roughly 7 pH. Okay? So there you go. You have to know that standard free energy doesn't occur naturally, right? I mean, not everything has a pH of 7. For instance, your blood has a pH of 7.4. So not everything is, picture perfect. So what I'm trying to say is standard free energy is manipulatable. It only occurs in lab experiments, right? This isn't realistic, so that's not realistic. It just offers some experimental values or data, okay? It's just used for comparisons. When we talk about catabolism and anabolism, what are we trying to say? Well, catabolism is when your body doesn't have enough fuel or, fats or proteins or whatever, so it starts eating itself. Well, not really eating, but it starts breaking down some cells in order for energy, right? So for instance, if a person is in the desert and they're lost, and they've been in the desert for 3 days with water, well, they don't have enough food, right? So they're depleting their carbohydrates, they're depleting their fat storages and their proteins. So the body says, hey, I'm not getting any calories, but I do have some muscles. It's not a lot, but I can break down these muscles and I can use the energy that I break down for fuel. And maybe that's going to help me out for a week, right? So catabolism is essentially the breaking down of big cells for energy, okay? So catabolism breaks. So breaking, you can say macromolecules, breaking macro molecules for energy. If catabolism releases energy, what can we call it? So we can say that catabolism releases energy. Is it an endergonic reaction or an exergonic reaction? We can say that it is an exergonic reaction. So we can say that this is an exergonic reaction. And you're saying, hey, that doesn't make any sense. My body isn't breaking down muscles. It's not breaking down anything valuable. Well, yeah, it is. Whenever you're burning calories, for instance, your resting metabolism, that's essentially your body breaking down sugars, breaking down a little bit of fats for fuel. That's how you get through the day. You can afford not eating for like 10 hours, right? You're going to be a little hungry, but it's not going to hurt you. That's just your body performing catabolism, right? So that is a favorable reaction. That's how your body survives every day. We can also talk about anabolism, right? So anabolism, that is anabolism. It's essentially the opposite. It's actually building macromolecules. So builds macromolecules. Right? And does that require energy or does it release energy? It requires energy. Does that mean that it's endergonic or exergonic? It's endergonic. Does that mean that it's favorable or non-favorable? It's actually not favorable. It's a good thing to build macromolecules. Obviously, whenever you get a cut, your body builds the platelets and it protects your skin. You have a scab, but whenever that scab falls off, you have a new, area of skin, right? So that is anabolism. Your body produced something. The reason why it requires energy is because you got to have energy to supply these cells with what they need, right? So you get energy from food, you get it from vitamins, you get it from the sun. It requires energy, and it's not favorable because, thermodynamically, you required something, you have more energy now than you did 2 minutes ago, so it's going to be endergonic, okay? Now, usually on exams, they're going to throw a trick question at you. They're going to say, okay, well, between catabolism and anabolism, which one releases heat? And you're saying, oh, catabolism, because it's exergonic, you're going to be releasing heat. Well, that is partially correct. The true correct answer would be both. Why? Well, catabolism, in order for you to break down macromolecules, you're going to have to go into, let's say, proteins, and break down the little baby bonds in the proteins. Whenever you break a bond, you can think of it as lighting a match, right? So you break a bond, and you have a little spark of energy, right? So if you light a match, a little spark of energy, you have some heat. Well, in anabolism, to build macromolecules, you're going to have to break little bonds and reform them into bigger bonds. And so whenever you break the little bonds, again, you're going to have some heat. And so in order to build or to break something, you're going to have to break down little bonds, no matter what. And no matter what, you're going to be releasing heat. Usually, when systems are not in equilibrium, that's for the most part how reactions are made, right? You're not going to have 3 chlorines and 3 potassiums in a system. No, it's usually like 5 chlorines and 2 potassiums. So whenever the system, or whatever you're trying to measure, is not in equilibrium, usually you're going to have to put an energy source, right? So you could put in like 2 joules of energy. But whenever you do that, that's going to affect the Gibbs free energy. So how do we measure that? Well, we can use this, right? So we can use this, right? There isn't a cool name for that. I wish there were, but there isn't. So we're just going to put that as the Gibbs free energy prime, or G prime, all right? So what is G prime? G prime is equal to the gas constant, so it's equal to the gas constant multiplied by the temperature, usually in kelvins, multiply that by the natural log, so ln, right, of the K equilibrium. So that's an equilibrium constant that's usually given in experiments or, practice problems or something, right? If you are just focusing on the concepts, you just got to know that everything is proportional. What I mean by proportional, I mean that everything it's connected. If the if the temperature went up, then Gibbs is going to go up. If the gas constant, if you change the gas constant, that's going to change the Gibbs free energy. If you change the K equilibrium, that's going to change Gibbs free energy, okay? Usually you don't have to worry about this one because that's a constant. That's always going to be a specific number. So there you go. The only thing that you can really change is this guy and this guy. All right. So what does that mean for us? Like, why should I care? How do I even put that into perspective? Well, let's say that you have a reaction, A plus B can give you this right here. So we have A plus B gives you C plus D. So now we have to find what delta G prime is. Okay, so what is that equal to? Well, we can say that delta G prime is going to be equal to what? Is going to be equal to the Gibbs free energy, so it should work on my triangles, right? Minus RT ln. Now remember what the K constant was. K constant is just, products over reactants. So that's okay. We can just do this. We can do the products, specifically the concentration of the products or the concentration of the reactants. Okay. So there you go. Wow, I didn't put my G. So that's really sloppy. But here we go. Right? So that's what's supposed to go there. Okay? In some textbooks you might see this as something different. So in some textbooks you might read this as this. You might have that Gibbs free energy is equal to G prime plus RT ln, and let me be lazy for a little bit. Okay, all they did was they just added this portion right here to this side. That's all they did. Personally, I prefer this one. So there you go. So it's saying that the total free energy is equal to the energy that you put in plus this this stuff. Okay. So what is that trying to say? Well, it's trying to say that in order for some reactions to occur, you're gonna have to put some energy in it. Why is that? Well, some reactions, they don't want to go into completion because overall it would be what? It would be an endergonic reaction. So sometimes you're going to have to put some reactions together, right? It's kind of like politics, right? Whenever somebody tries to pass a bill, a reaction, they're going to say, well, I don't want to do this because it's going to increase taxes. But then some other politicians are going to say, okay, well, what if we added so-and-so? What if we did this as well? What if we, did a compromise for you? And the person's going to say, okay, well, if you add that to this bill, to this reaction, I'm going to pass it. So what I'm trying to say is some reactions do not want to go into completion because it's not favorable. And so you have to add something to it, preferably a different reaction, in order for the total reaction to be favorable, right? So that is what this this equation is trying to say. Okay. So overall, you have to add something to this equation for the reaction to become exergonic. Well, the reason why we're focusing on thermodynamics is because it will actually allow us to predict if a reaction is going to go into completion. So how do we do that? Well, we can do that by doing some advanced calculus, right? So we can say that the total G prime, the total free energy, is going to be equal to this guy right here. So the total free energy of reaction 1 plus the total free energy of reaction 2. And I'm just adding the primes because we're doing this experimentally, okay? Under perfect conditions. And you can just do this into you run out of reactions, okay? So really advanced high school stuff. Ooh, spooky, right? I mean, it is October, so we're gonna get into some very spooky stuff, right? So what does this mean for us? It means that if we add up the total energies from these two reactions, we can actually find if the overall reaction is going to go into completion. But before that, we can solve it like a puzzle, because you don't want to be mixing reactions together. You just want to have a complete equation. So we can say that, well, if a product shows up as a reactant later on, we can cancel it out, right? So it's kind of like doing math. Well, let's see, does A show up anywhere? No, it just shows up here. What about B? No. What about C? No. What about D? Well, D is a product, and D also comes out as a reactant. Okay, so we can get rid of that, right? So we can say that D can be eliminated. So we can do this right here and do that. So now everything cancels out, and we can write it as a standard equation. We can say that A plus B plus E is going to give us C. Let me redo that because it does not look good. There we go. C plus F. All right, so that's the overall reaction. Now, will that reaction go into completion? Let's find out. We can say, let's just do this right here. We can say that the total free energy is going to be equal to -3 kilojoules per mole of reaction 1 plus -2 kilojoules of reaction 2. And this is per mole. So -3 minus 2 is -5. So we can say that the overall standard free energy of this reaction is going to be -5. So this is -5 kilojoules per mole. And what does that mean? It means that it is an exergonic reaction, that it is spontaneous, and that it will go into completion. So that's all you have to do for those problems. If you see that on a exam, well, you just do simple math. You just add up the standard free energies, bada bing, bada boom, you're done. Pretty simple. I love this. Right now, in your body, you have this molecule floating around. It's just floating around. Why do we have that? Well, this is actually an ATP molecule. The reason why we have ATP molecules is because that is the currency that our body uses to do reactions. So for instance, you have a sodium-potassium pump in your body that helps regulate the action potentials in your membranes. Action potentials send messages in your brain to your nerves and stuff. So to actually create action potentials, you can use ATP. How do we use ATP anyways? Well, you essentially need water, and your body's like 70% made of water, so you have a lot. And water is going to do some reactions right here, and it's going to essentially cleave off this guy right here, okay? That's what it does. So whenever that happens, you're gonna have ADP. ADP. So that's adenosine diphosphate. So you only have 1 and 2 phosphates left. Okay, so that's di. Right now you have 3, so that is why it's called triphosphate, right? So whenever that bond breaks via a process called hydrolysis, right? So this is hydrolysis. So water, hydrolysis. Lysis is cut, water cut. That's what it's doing. Whenever this happens, you're going to actually be releasing about -31 kilojoules of energy. So that is a massive amount of energy. So that is why if you have a chart like this, and it's for ATP, you have ATP does some reaction and does that. So you know that the products is going to be at a less energy than the reactants. So that's going to be what? It's going to be an exergonic reaction. It's going to be spontaneous. Your body wants to be cutting these molecules, okay? So it is a very efficient and economical way to create energy within your body. Is that catabolic or anabolic? It's actually catabolic, right? So your body wants to do that, okay? So that's pretty cool. And now we can talk about electrostatic repulsion against the phosphate atoms. So that's a big word, but it's saying, all right, so aside from the energy, why in the world would your body want to get rid of this molecule? Why would it happen? Well, notice that you have a bunch, a lot. You have like a billion, no, it's like actually 7. You have like, you have 7 oxygens attached to like these phosphates, right? So that's a lot of electrostatic repulsions. So because you have a lot of atoms that have electrical charges, they don't want to be close to each other. They're kind of pushing and shoving each other. It's like going on a road trip with a family. You're going to be clustered inside a van. You don't want to be there. You just want to leave. So whenever you get to a gas station, you can stretch your legs. That's essentially is what happened to this phosphate group. The phosphate wants to be cut out of the molecule. It wants to go away. It wants to go do its own thing because it's too clustered. It's too hindered, right? So there's a lot of energy right here. It doesn't want to be here. The water cuts it and it's free. It's free to go and it's happy. So because of electrostatic repulsions, this process is going to be favorable aside from the energy released, right? So that's usually something on exams. They're saying, you know, name like 3 reasons why we would want to cut ATP and form ADP, right? So there you go. You know, the products are more stable, meaning that they're just going to kind of rejoin with other molecules, and that's always a good thing. Your body wants to recycle molecules together. So if you make a product that is stable, you're going to want to keep doing that reaction, right? So a singular phosphate ion So we can just put pi. That's just this guy right here by itself. That's stable. Like it's going to do some other reactions in the body, right? So that's also pretty good. Okay. Now we can talk about ADP. ADP also has a lot of energy in it because you still have 2 phosphates. So this can actually be broken down again to make, you can make AMP. So that's adenosine monophosphate, and again, another phosphate ion. So this is also pretty good. It has a lot of energy in it as well. So has energy, has tons of energy. And before I forget, ADP is actually stabilized by resonance. Okay, so if you were to look at this, you can actually say that, well, I can have different areas where I can put this double bond at, right? I could put it over here, I could put it over there, you know, and because I have more options to put the resonance structure, I am stabilized. Okay, so that's another reason why we want to break down ATP. So to summarize, ATP releases a lot of energy. You don't have to know the numerical value. You just know that it releases a lot of energy, so it's going to be exergonic. Initially, there is a lot of electrostatic repulsions. It doesn't want to have that, so it wants to be cut so it can limit the amount of electrostatic repulsions, and the product is going to be resonance stabilized. Okay, so ADP and the phosphate ion are resonance stabilized. So those are the main 3 reasons why we want to cut ATP. Now we're going to be covering thioesters. So remember that thio, so let's say thio is actually going to be over here. So let's move this, obviously. Let's do this right here. So thioesters actually do this. So we have thio, it means sulfur. Okay, so you have sulfur, and then an ester means that we're going to have essentially a double bonded oxygen right here and some other oxygens, which you'll see. But what I'm trying to say is thioesters actually release a lot of energy. They actually release more energy than ATP. So thioesters release more energy than ATP. Okay? So if you want to get into a specific number, it's about negative 31.5 kilojoules per mole right here. Okay? And all you need to look at is, does your molecule have a thio in it? Well, here we have acetyl, right? Coenzyme. So this is well, actually not acetyl coenzyme, but this is just coenzyme A, right? So we have coenzyme A, and we have the sulfur and hydrogen structure, right? So this guy right here is going to be reacting with an acetyl, and you'll see that in this section right here. So we have this molecule. It has the SH. Co-A is just everything that side, okay? So don't focus on the big chain. It may look scary, but it's not. So we have coenzyme A, and we can have the what? We can have it reacting with the acetyl. Whenever it reacts with the acetyl, it's going to make acetyl coenzyme A. And you might actually have heard about that in other biology classes. Acetyl coenzyme A is really, really common in metabolism. So this is common. So common in metabolism. And you can tell why, because in metabolism you need a lot of energy in order to break down molecules and do reactions, right? And so if I break down this this bond right here, you're going to be releasing about negative 31.5 kilojoules per mole, or you can just say a lot of energy. Okay, so that is usually how it works. Okay, so we can say that for trivia, this is actually derived from vitamin B5. So if you have an exam, you can say that coenzyme A is just made from vitamin B5, right? So you should always get your B vitamins in your diet. So to summarize, thioesters release more energy than ATP molecules. Thioesters is essentially any molecule that has the SH bond, okay? And then you're going to be reacting it with an acid, okay? You're going to be reacting it with an acid, and you're going to have this thioester bond. So if you break that bond right here, you're going to get negative 31.5 kilojoules per mole, and that is a very, very large amount of energy. It is most often found in coenzyme A, and coenzyme A is normally found in the metabolism. So in metabolic reactions, you're going to be using coenzyme A to do the reactions. So in the very beginning of the video, we were talking about catabolism and anabolism. So catabolism and anabolism. So we'll just put anabolism right here. Actually, anabolism right here. So what are the differences? So this one right here is a degenerative process. Okay, so it's going to degenerate molecules. So degenerative process. Okay, so it's going to be releasing energy and it's going to be creating some molecules. So it's weird, right? But whenever we're talking about cellular metabolism, you should know that NADH, FADH2, and ATP. Okay, so they actually make, well, this process actually makes NADH, FADH2, and ATP. You're probably wondering, well, how does ATP get created? I thought we break it. Well, ATP is actually a product of NADH and FADH2. Okay, so these guys right here, they make ATP, and you make those guys from catabolism. All right, so anabolism is actually the creation of molecules, right? And this actually uses the products of catabolism, right? So we can type that. Say uses NADH, so NADPH. It's really weird. So right here, that's phosphate. So it's going to be doing some other reactions, but ultimately it's going to be using NADPH. Okay, so it uses that. Also uses FADH2. And ATP. Okay, so that's all you really have to know for like the big picture, right? So you should know that catabolism is a degenerative process, and it creates NADH, FADH2, and ATP. Anabolism creates molecules, okay, because it's a regenerative process, and it uses up the NADPH that just does some other reactions to become the phosphate, and it uses FADH2 and ATP. When we're talking about metabolic pathways, we're talking about specific order of sequences, right? So the most famous metabolic pathway would be glycolysis, right? So you have reactant A plus reactant B gives you reactant C, right? You just can't you can't go from A to C, right? No, you have to go to a different step, right? So this is a sequential metabolism, right? So you go from step 1, step 2, step 3, step 4, etc. And then ultimately you're going to get a product. You cannot be skipping steps. That's not how it works, right? And in this class, we're going to be focusing on carbohydrates and lipids, right? So we already covered lipids. Carbohydrates is going to be covered later on, but we're going to be focusing on those metabolic pathways. Unfortunately, nucleotides and amino acids are not typically covered in introductory biochemistry, so that would not be covered. Okay, so yeah, essentially energy. Let's move on. So energy that is created is usually in the form of ATP, right? So you want to be forming ATP. Whenever you're doing glycolysis, you do some series of reactions, and your product is going to be like 4 molecules of ATP. Okay, so that process is actually catabolic. That's weird, right? Well, think about it. You got one molecule right here, and you had another molecule, and you said, well, I'm going to break these apart, and then I'm going to make ATP. So let's just say that molecule C is ATP, okay? So overall, the process is catabolic because I was breaking down molecules. The formation of this guy right here is technically anabolic, but overall, the reaction is catabolic. This makes sense. Your body, whenever you're running, you're using your sugar storages. You're not making sugar storages, right? You're using your sugars to create ATP in order to feed your body, right? To feed your muscles. And so that is a catabolic process, right? So yeah, ATP is not stored in the body. It's actually used in the body, okay? So ATP is the main unit of energy, and you do not store it. So you have fat storages, adipose tissues that you store fat in, but you do not have tissues that are specifically there to hold ATP. That's not a real thing. If it was, that would be amazing. You would be like a super athlete or something. You could probably do like the Olympics. That would be cool, right? If that happens, you should like totally sponsor me. So ATP is used and then it is remade, right? So whenever we use ATP, we break it down to ADP, and then that molecule of ADP can go through a series of metabolic processes, and it can reform the ATP molecule, okay? So that's why we don't store it. We could just recycle. So we reduce, we reuse, and we recycle, okay? So that's good. We do that. So ATP, we can say that ATP right here, ATP is common to both anabolic, or not anabolic, but anaerobic and aerobic pathways, okay? So say aerobic and aerobic, we can say organisms or pathways, okay? Because sometimes depending on the pathway that you're using, you will still be making ATP, okay? So that's what it is, right? Okay. So like I was saying, metabolic pathways are actually very, very in-depth, and they happen in stepwise procedures, okay? So we have step 1, we go to step 2, we go to step 3, and let's say we go to step 4, right? And step 4 gives us the product. Well, this whole series of metabolic reactions, these steps, are very, very regulated, okay? So in each step, you're gonna have enzymes, and those enzymes are gonna be reacting with substrates, right? So let's say enzyme A reacts with substrate B, and that gives product A. And then the product A is gonna be reacting with enzyme B to do the reaction all over again and kind of propagate, to move in the reaction sequence, okay? So we don't want to be messing up. You do not want the enzyme reacting with the wrong substrate. You do not want the enzyme to be inhibited if it's a good reaction. You don't want it to be inhibited. And so you have a lot of checkpoints in the reaction to make sure that everything is going good, everything is going fine. It's going to say, well, enzyme, you didn't react with the substrate, you're not making product, that's not good. We're going to have to insert more substrates. So your body sends messages to the brain, and the brain produces signals that make more substrates, right? So essentially these checkpoints try to give everything that is possible to do this reaction. So if you need more substrates, they're going to sense it, they're going to produce more substrates. If you need more enzymes, they're going to sense it and they're going to produce more enzymes. And so what I'm trying to say is that the metabolic pathway is a very, very regulated order of sequences, okay? So it has checkpoints, it wants everything to go very well. If that does not happen, the product is not going to form, and that could be lethal at some points, okay? So yeah, that was regulation. We will now be focusing on the catabolic pathway. So for instance, this is a good example of kind of like glycolysis. Very, very, it's a big picture, okay? Because glycolysis, which we're going to be covering later on in a different chapter, is very in-depth. There's a lot of steps, but this picture essentially summarizes it. Okay, so you can copy it down for later reference. Well, what I'm going to explain to you is that this whole process of glycolysis is catabolic, because overall we keep breaking down molecules to make a product, right? So we're releasing energy, which is good, and we're going to make a product, which is also good. And what happens here is that in step one, because of course metabolic pathways or step-by-step procedures, in step one, essentially we're just breaking down macromolecules into the building blocks that they are. Okay, so let's say that you ate some chicken. Okay, you ate some chicken, and your body is going to start breaking down those muscle fibers and those protein fibers into their building blocks of amino acids. Okay, so now you have alanine, you have leucine and stuff in your body. So that process was catabolic, right? And you're going to do the same thing to glycogen and starch. You're going to do that to your fats like triglycerols, right? So essentially anything that you eat is going to be broken down to their building blocks atomically. Okay, so step one releases little to no energy. So releases little to no energy to no useful energy. So this process, step one, this energy is not really good. It's not useful. It's like 0.001 kilojoules. It's small, negligible. Okay, so that happens. But now you have all these building blocks and you want to make a product. How do we do that? Well, your body says, well, I got all these building blocks. If I convert them to just one building block, I can use that one building block to make a product. So it says, okay, how can I convert amino acids into something common? How can I convert glucose and fatty acids into something common? What we have now is, drumroll, we have acetyl coenzyme A. Okay, so we just covered acetyl coenzyme A, and I told you it's very common in metabolic processes. And what your body does is that it's saying, okay, I'm going to get my amino acids, my glucose, my fatty acids, I'm going to convert them all to acetyl coenzyme A. And then from there, I can make acetyl coenzyme A into my product, which is going to be ATP, ultimately. Okay, so step two is convert to common product. So I'm just using glycolysis as an example. Okay, so these steps are just in general. Okay, so step two, convert to common product. Okay, so there you go. In step two, we're going to convert to a common product, and then step three, we're going to perform an oxidative reaction. Okay, so oxidative reaction is what is going to give us a NADH and FADH2. Okay, so this right here, acetyl coenzyme A, is going to do some oxidative reactions. Okay, so yeah, usually catabolic processes are oxidative. Okay, so I'll cover what oxidative is later on. So yeah, there you go. So I want to mention that in your body you have cells, obviously, but in those cells you have different organelles. So for instance, you have the mitochondria, you have lysosomes, you have ribosomes, nucleus, etc. Well, some organelles can individually do metabolic pathways. Why do we have that? Well, we have that because you don't want just one cell, right, doing a metabolic pathway. You don't want that. It takes too long. If you have one cell that has 10 organelles inside of it that can do the same metabolic pathway, is going to give you more product in a shorter amount of time. Okay, so I mean, it's faster, it's more efficient. And another reason why we do that is because some pathways, you want another pathway to balance it out. So let's say that you make a lot of product A, okay? And your body says, okay, well, I don't want too much, so I'm gonna make another pathway, and we're gonna make product B. And product B is going to eliminate product A, okay? So in this cell, we're going to be having little tiny organelles, the little factories that produce product A. But then I'm going to produce other organelles that kind of balance it out, so I don't have too much of a good thing, right? Because that would be pretty bad for the environment. And so we have kind of like these parallel metabolic pathways in our body, okay? And that allows us to balance it out. So don't forget that, okay? And if you want to relate that to the bigger picture, you can consider the anabolic pathway to be the counterbalance to the catabolic pathway, right? So if your body just kept breaking down macromolecules for energy, you would die. So your body says, okay, well, I need to make an anabolic pathway so I could regenerate some macromolecules, okay? So if I have too much anabolic pathways, I'm going to be building a lot, and I'm going to be using all my energy, and I'm going to die. So to counter that, the catabolic pathway will be created in order to release some energy, okay? So there you go. So now we'll be talking about oxidation reduction reactions, or typically called redox reactions. So some redox reactions. This is shorthand for reaction. So what is a redox reaction, and why is it important? Well, catabolic pathways, which is what we're talking about right now, are oxidative reactions, okay? So catabolic pathways, catabolism, is an oxidative reaction, okay? So essentially amino acids, monosaccharides, and lipids are oxidized. And for shorthand, we're just going to say oxy, okay? So oxy means oxidized, right? So what does that mean? Well, oxidize means that they lose electrons, okay? So that means that they lose electrons. Let me write it over here. So oxidized means lose electrons. So say lose electrons. We'll just say E negative, right? So there you go. That is called, that's what it means to be oxidized, right? But when something is oxidized, what do we call that? If something is oxidized, we call that a reducing agent. It's really dumb. So if something is oxidized, is oxidized, it's called a reducing agent. Right? Okay, so how do we have something that is easy to remember? What do we have? Well, we have the mnemonic oil rig, right? So we have oil rig, and I'm running out of space, so I'll just put it over here. Oil rig. Okay, so if something is oxidized, so oxidation, oxidize, is the loss of electrons. So this is reducing agent. Okay, so yeah. Let's just rewrite that. So what I'm trying to say is oil rig, oxidized means it is the loss of electrons. So when I said that catabolism is an oxidative reaction, I'm saying that in catabolism we're going to be losing electrons. Amino acids, monosaccharides, and lipids are just going to be losing electrons. And we can say that these are going to be reducing agents, okay? So we can say that these are reducing agents. And specifically when we're talking about monosaccharides, we're going to be calling them reducing sugars because they're going to be reducing certain things inside the reactions. So what is RIG? Well, we can say that reduction reduction is gaining electrons. And we can say that this is going to be oxidative or oxidating reagents, okay? So we can say oxidizing agents. So what's an agent? An agent is just a molecule that does something in a reaction. So if I have molecule A and it's a reducing agent, is going to be what? Is going to be losing electrons in this reaction, okay? So we can say that molecule A was oxidized and therefore it is a reducing agent. So my tip for you is that whenever you're working in reactions, just say the opposite. You're saying, okay, well, molecule A was oxidized, meaning that it lost electrons. Since it was oxidized, I'm going to do the opposite. It is called a reducing agent. Okay, molecule B gained electrons, so oil rig. Okay, so it gained electrons. That means that it was reduced. If something is reduced, we think of the opposite. It is going to be the oxidizing agent. Okay, so there you go. For every reaction that occurs, there's always going to be half of the reaction being a reduction reaction, and then half of the reaction being an oxidation reaction. Yeah, it's very fun to say. What I'm trying to say is, if you have a coin, half of that coin is going to be a head, and then half of the coin is going to be the backside, or the tail, right? So in a typical reaction, half of it is going to be reducing, and the other half is going to be oxidating. And essentially, they balance each other out. Okay, so we don't have too many things reducing and not enough oxidation, right? So you're probably wondering, okay, well, why is reduction unfavorable? Well, what is reduction? Okay, well, oil rig, right? So oil rig, and I think we should move into some renewable energy sources, not just oil, by the way. So oil rig, reduction is gaining electrons, and we know that this is going to be an oxidating agent, okay? So I have my oxidating agents right here. They're just being reduced. Well, it says gain electrons. Well, if I put electrons into a system, there's going to be more energy in the system. So electrons are like little tiny baby particles that fly around really, really fast because they're light, right? They're very lightweight. So it's kind of like throwing a bunch of kids into a room in a pre-K and then throwing like a bucket of candy in the room. So all these little kids are going to be gaining electrons, right? So my little oxidating agents, the toddlers are going to be gaining electrons, gaining candy, and they're going to be higher energy. They're going to have a higher energy state. They're going to be running around, crawling on the walls. One of them is going to be on the ceiling doing like the little exorcist neck thing, right? So so it's a higher energy, okay? And they're going to be reduced. They're going to be oxidizing agents. They're going to be reduced, and that means they're going to be gaining electrons, gaining energy. So it's this one gains electrons and gains energy. Okay, so never give candy to children without permission. Okay, because they're going to be really hyper, and some parents don't want to deal with that. So that's why this is unfavorable. If you're gaining energy, what are you doing? Is it going to be endergonic or exergonic? It's going to be endergonic. Okay, so we can say that this is endergonic. And we can also say that for a reduction, the standard is going to be what? Positive or negative? It's going to be positive. And we can just do the opposite over here. If this is oxidizing, what is oxidizing? Well, it means oil. Oxidizing is loss of electrons. So now those kids, after running about 10 miles in the room, they ran out of energy. They ran out of candy energy or electrons, and now they're going to be having a sugar crash, right? So now they're tired. They're going to be losing energy. They have become reduced. Now my oxidizing agents are going to be reducing agents, okay? So they're reducing their energy, and they're going to be sleeping. So we know that this is going to be reducing agents. So over here we can say this is oxidizing agents. And we can say that this is going to be reducing agents. I'm not saying this is a reducing agent. No, I'm just saying that in general, your reducing agents are on the left side, or right side, excuse me. Okay, so oil rig, this is going to be my exergonic, because since I'm losing electrons, I'm losing energy. That's a good thing. This is going to be exergonic. And that means that the standard free energy, or G prime, is going to be equal to a negative number, right? So that's going to be overall very positive. So remember how I said that sometimes you have to mix reactions in order to get a overall negative reaction? That's what we're doing here. In the beginning, we have a lot of reactions that are not favorable. They're all positive. But if we combine them together, overall, they would give us a product that is spontaneous, that is negative, right? So if I add all my non-favorable reactions, I will produce a reaction that is favorable, that is negative. Okay? So yeah, there you go. We can talk about kind of like molecules that are being reduced or oxidized. So here are some general rules to think of. So if oxidized, you can tell because you actually have less hydrogens. So we have less hydrogens. Okay, so yeah. And because we're really focusing on this one, so we want to lose electrons, right? You can consider hydrogens to be electrons. I know they're protons, but let's just pretend because it makes it more simplified, right? So if you're oxidized and you look at a molecule, so let's say that you have a before and after, right? Let's say over here we have CH3 and I go to CH2 and I ask you, okay, was this oxidized or reduced? Well, you're going to say, okay, well, oxidized means to lose something. So I lost a hydrogen. That means that I was oxidized, right? Okay. You can also consider it if oxygen bonds increase. So if I go from this guy right here and I do this, right, to make a carbonyl, well, we can say that that was oxidized. Literally, the amount of bonds to oxygen has increased. And we can actually do the opposite for reducing agents, okay? So for reducing agents, or sorry, for a reduction, if reduced, well, then you're going to be gaining hydrogens. Okay, so if I go from CH3 and I go to CH4, we know that this is going to be what? This is going to be a reduced molecule. So we know that this is an oxidative agent, right? So if you gain hydrogens, you're going to be reduced, right? And likewise, if you have less bonds to oxygen, you're going to be reduced. Less bonds to oxygen. So excuse me, so we go oxygen. So if we go from something like this guy right here, and we go to this guy, we know that we were reduced because the amount of bonds to oxygen was reduced. And so this video is kind of dragging on for a bit. I do not want to take up a lot of your time, so I'm going to be cutting the video here. Okay, and we'll continue cellular metabolism in part 2. So I hope that you understand catabolism, anabolism, standard free energy, Gibbs free energy, and it's kind of like the basics of cellular metabolism. I hope you do well in your exam, and thank you so much for watching my videos and for spending your day with me. So have a great day and be safe. Love you.